In this topic, we will be looking at inheritance programs that are more complicated than those seen in garden peas. We will be looking at degrees of dominance, the relationship between dominance and phenotype, the frequency of dominant alleles, multiple alleles, pleiotropy, epistasis, polygenetic inheritance, nature-nurture, and integrating a Mendelian view. Mendel picked a good model for studying inheritance. In the 7 traits or characteristics he chose to study, the dominant traits had complete dominance. Other plants show incomplete dominance. Each allele contributes to how the flower will look, rather than one completely covering the other one up. Take these snapdragons. The allele for red codes for a red pigment in the petals. The white allele is a damaged allele, so no pigment is produced. If you have 2 red alleles, you get a double dose of red pigment and the flower is red. If you have 1 red allele and 1 white allele, the flower produces half the amount of red and the flower is pink. In the F2 generation, you get 1/4 red flowers, which are homozygous red. You get half pink, which are heterozygous, and 1/4 white, which are homozygous white. You can see how this would be confusing when we were learning the basics of genetics and didn't know about genes and chromosomes yet. But since we do know the basics and we can watch chromosomes separate using a microscope, it makes sense. Let's look at a human example of incomplete dominance. Tay-Sachs disease is an inherited disorder in which the gene for an enzyme for breaking down worn-out lipids is damaged. The damaged gene can't code for the needed enzyme. Lipids build up in all of the cells of the body, but the cells of the brain are more sensitive to this than, say, muscle cells. The affected child suffers blindness, seizures, and increasing mental and motor function dysfunction until they die. It's a terrible disease. Individuals that are heterozygous for Tay-Sachs gene do not develop Tay-Sachs disease. They do produce half the number of enzymes as individuals who are homozygous for the non-Tay-Sachs gene. We are like the snapdragons in this regard, but rather than having 2 different colored flowers, we show different levels of enzyme levels that can be determined by biochemical tests. This shows a relationship between dominance and the phenotype. An allele that is called dominant because it is seen in the phenotype, not because it somehow subdues a recessive allele. Let's go over the terms genotype and phenotype. Genotype are the alleles you have. Phenotype is what is expressed. Having 1 or 2 alleles for Tay-Sachs is genotype. Having the disease is the phenotype. We tend to think of phenotype in terms of eye color, hair color, skin color, but it's also how well your liver functions or the ability to produce digestive enzymes. One would think that the dominant alleles would be more frequent in a population, but that isn't necessarily so. Going back to our Tay-Sachs example, Tay-Sachs disease is more prevalent in Eastern Europe than in the United States. There are more recessive alleles circulating in the population. It has to do with the alleles that are available to resort through marriage. Let's look at another example that leads into the next slide. Type O blood is recessive. You have to have 2 copies of the O allele to have type O blood. The recessive O allele is very common in the United States, leading to more people having type O blood than any other blood type. So, in the United States, the O allele is more common than the other dominant alleles. This is not the case in other parts of the world, by the way. Speaking of blood, we have more than 2 alleles for blood type or blood group. We have alleles for A, blood carbohydrate, we have alleles for B, blood carbohydrate, and we have alleles for no blood carbohydrates or O. In a population, you can have multiple alleles, but in a diploid individual, you can only have 2 copies of that allele. This is a good spot to talk about codominance, or where both alleles contribute to phenotype. If you have the allele for A and the allele for B, you're going to have AB blood. Both carbohydrates are expressed, and we can detect them with biochemical tests. If you have the allele for A carbohydrate and the allele for no blood carbohydrate, or O blood, you're going to have type A blood, since A is completely dominant over O. Confused? It's not too bad. Think about how the alleles are expressing proteins, and pause the video to go over the diagram more closely, or go over the diagram in the textbook. And you can always ask me to go over it again in class. Let's go over another term, pleiotropy. This is where one gene causes multiple effects in the organism. Humans have two examples of this. In cystic fibrosis, a gene for mucus protein produces extra thick mucus. It is hard for the cilia in the body to move it out. It causes problems everywhere in the body that there is mucus. The lungs become clogged with mucus and the affected person has a hard time breathing. The intestines become clogged with mucus and the affected person has a hard time digesting their food, and so on. Sickle cell disease is another example. A mutation in the hemoglobin results in red blood cells forming a comma or sickle shape when they're in the presence of high levels of oxygen. These cells are rigid and get stuck in the capillaries. When capillaries are blocked, that damages the organ or muscle or brain. Anemia can also result as the sickle cells are cleared out by the spleen, which is also not happy to see all those damaged red blood cells. In both cases, one gene causes effects in multiple organs or organ systems. So far we've talked about how different versions of one gene can affect phenotype, but most characteristics are caused by more than one gene. Let's take fur color in mammals. One gene in mice codes for the pigment of the fur. Another gene codes for a protein that moves the pigment out of the cell that made it into the shaft of the fur. Mice have two alleles for pigment, black and brown. Black is dominant over brown. But they have two alleles for the transporter gene. If they have allele that codes for a non-functioning transporter gene, the pigment is not moved into the fur and the mouse is white. This is different than albino, by the way. When one gene inhibits or helps another gene, we call this epistasis, from the Greek for standing upon. Think of the non-functioning gene as standing on the functioning pigment gene, keeping it from being expressed. So looking at this Punnett square, we can see that the two genes separate independently, and we get a lot of non-parental phenotypes when we cross the heterozygotes for both genes. Don't despair if it's difficult the first time through. Dihybrid crosses are difficult, but repetition and practice make it easier. Now let's talk about human skin color, since we don't have fur. At last count, there are 19 genes that all contribute to one human characteristic, skin color. We call this polygenetic inheritance, or having multiple genes that affect one characteristic. And this is why humans make such terrible beginning models for studies in inheritance. When you look at the polygenetic characteristics like skin color, height, and so on, it's far too complicated. Peas, with their one gene affecting flower and seed color, are much more simple. And if you look at this chart here, they're only looking at 3 genes. If we looked at all 19, we wouldn't be able to fit this Punnett square onto this slide. But it gets more complicated from there. Environmental conditions affect the expressions of genes that cause different phenotypes. I know several people that have bought hydrangeas, which are the flowers in these pictures, and have been really mad when the new flowers aren't the same color as the lovely pink or the blue that they picked out in the store. With hydrangeas, the chemical composition of the soil determines the color of the flowers, not necessarily the genetics. The soil of their garden was different from that of the nursery. By the way, nursery workers should let you know when you purchase plants that react this way. But humans are similar. I may have genes that code for tall growth, but if I grow up during a famine, I'm going to be short. I don't have the energy for my cells to lengthen my bones and muscles. Like how I work chapters 9 and 10 in. Once I reach a certain age, about 19 in women and 21 in men, the genes for lengthening bones and muscles will turn off and I'll be stuck at my height, no matter if the famine is over or not. So let's look at how genotype can result in phenotype. I have a lot of genes, from A through B, clear through to X, but I'm only going to put 3, just to reduce the complexity. Gene A affects genes B through X. Gene B affects gene A and genes 3 through X, and so on. My genotype, or all the alleles for all my genes, affect each other. But it doesn't stop there. There are a lot of environmental factors that express gene expression. Do I live in a city with a lot of pollution or a green city with little pollution? Do I live in a rural area where I engage in heavy lifting for long hours, or do I spend hours hunched over a computer keyboard? Am I a man or a woman? That affects what genes are turned on and when, and on and on. All of these different genes and environmental combinations combine to create the phenotype or the unique individual that you are. And you are continuing to change throughout your life. Well, that's it for this concept. It was probably enough, huh?